Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine dealing with something super common like bacterial vaginosis, BV, and the usual treatment, just doesn't cut it. Over half the time, it doesn't even work.
Yeah, it's really frustrating for a lot of people.
But what if there was a way to look at the DNA of the bacteria causing your BV and figure out the best way to treat it?
Well, that's what we're diving into today. This research is looking at how to personalize BV treatment.
That's so cool. I'm excited to learn more. So BV, it's a pretty common thing, right?
It's super common. It's basically when the normal balance of bacteria in the vagina gets disrupted, affects something like 1 in 3 women of reproductive age here in the U.S.
Wow, that's a lot of people. And I know it can cause some not so fun symptoms like odor and stuff.
Definitely. The sources we looked at mentioned things like distinct odors, potential discomfort, and even possible links to other health issues down the line.
Yikes. Okay, so not something you want to just ignore. And you mentioned the usual treatment isn't always effective.
Yeah, the first-line treatment is usually an antibiotic called metronidazole. It works for some people, but the data shows it's ineffective for over half of the women who take it.
So they go through the treatment and it just comes back.
Exactly, and that can be super frustrating and really impact someone's life.
For sure. So this new research, it sounds like it's offering some hope.
Definitely. Researchers at Drexel University have been looking at the genetics of the bacteria involved in BV. They've actually developed a new test that can pinpoint the specific groups of bacteria causing the infection.
Wait, so they can look at the DNA and say, okay, this type of bacteria is causing your BV.
Exactly. And knowing that allows doctors to potentially prescribe the most effective antibiotic for that specific type of bacteria.
That's amazing. No more guessing games with the antibiotics.
Right. It's a move toward personalized medicine for BV, which is super exciting.
So in this deep dive, we're going to break down how this test works, what it tells us about BV, and what it could mean for you in terms of getting better, more personalized treatment.
Sounds good.
Awesome. We've got a couple of sources for this deep dive, a recent health news article, and a really in-depth scientific paper published in the journal Genome Medicine.
Great sources.
Yeah, so let's start with the study itself. What were the researchers trying to find out?
Well, they were really focused on understanding the genetic differences within Gardnerella vaginalis and similar bacteria.
Gardnerella vaginalis, that's the main bacteria linked to BV, right?
Exactly. And they were specifically looking for genetic links to why some of these bacteria are resistant to metronidazole, the antibiotic we talked about.
So they were trying to figure out why the antibiotic doesn't always work?
Exactly. Their goal was to develop a test that could use this genetic information to predict which antibiotics would be most effective for each person.
Okay, so they were going beyond just knowing it's Gardnerella vaginalis causing the BV. They wanted to get more specific.
Right. And this actually builds on previous research that had shown that Gardnerella vaginalis isn't just one single type of bacteria, but actually a group of closely related bacteria.
So there are different flavors of Gardnerella vaginalis.
You could say that. And this study took that idea even further, creating a super detailed family tree of these bacteria to really understand their relationships.
A bacterial family tree? That's cool. But how do you even go about making one of those?
Well, they started by looking at the entire genetic code, the genome, of 129 different strains of Gardnerella bacteria. They use this technique called whole genome sequencing.
Whole genome sequencing. Is that reading the entire DNA blueprint of each bacteria?
Exactly. It's like reading the entire instruction manual for each strain.
Yeah.
And then they used a method called comparative genomics to compare those instruction manuals and look for similarities and differences.
Wow, that sounds super complicated.
It is. But from all that data, they were able to identify 11 distinct groups within Gardnerella, which are called genospecies.
11. So it's way more diverse than just one type of bacteria.
Right, and these genospecies then fell into even larger groupings called clades.
Okay, so we've got genospecies, those are the individual flavors of bacteria, and then clades, which are bigger groups of those flavors.
Precisely. And the important part is that some of these genospecies behave very differently when it comes to antibiotics.
Ah, so some might be resistant to metronidazole while others aren't?
Exactly. To figure that out, they took 60 of these strains and tested how well metronidazole could stop them from growing in the lab. They did a similar test with 63 strains using another antibiotic called Clindamycin.
So they were testing out which antibiotic worked best against which strain.
Yep. And they measured something called the minimum inhibitory concentration, which is basically the lowest amount of antibiotic needed to stop the bacteria from multiplying.
So like the minimum dose to knock out the bacteria.
Exactly.
Okay, so they've got all this genetic information, and they know how much of each antibiotic it takes to stop each strain. What do they do with all that data?
The next step was to connect the genetic information of each strain with how it responded to the antibiotics. They were looking for patterns, like specific genes or variations that were always present in the resistant strains. That helped them build their diagnostic tool.
So they were like, okay, all these resistant strains have this gene in common, so if we see that gene, we know it's probably resistant.
Exactly. And then they created these short DNA sequences called polymerase chain reaction-based primers.
Polymerase chain reaction, that sounds familiar.
You might know it as PCR. It's a technique used to amplify DNA so you can study more easily.
Right. Okay, so these primers are used to identify specific genospecies based on their DNA.
Precisely. They're like little genetic flags that say, hey, this is genospecies number 3, or whatever.
That's so cool. So after all that work, what were the key findings of the study?
Well, first of all, they confirmed that there's a ton of diversity within Gardnerella, solidifying the existence of these 11 unique genospecies.
Like way more than we initially thought.
Right. But even more importantly, they found that 2 specific clades, which included 5 genospecies, were completely resistant to metronidazole.
Completely resistant, so the antibiotic wouldn't have any effect on those at all.
None at all. But here's the interesting part. Those same strains were all susceptible to clindamycin, the other antibiotic they tested.
So even though they were resistant to one antibiotic, they were vulnerable to another.
Exactly. And the new PCR test they developed can actually distinguish between these resistant strains and the others.
So doctors could use this test to see if your BV is likely to be resistant to metronidazole right off the bat.
Yes, no more prescribing an antibiotic that's likely to fail.
This is huge, like potentially life-changing for people with BV.
I agree. It's a big step towards personalized treatment.
Okay, let's talk about what this could mean for you, the listener. What are the practical implications of this research?
Well, the biggest thing is the potential for truly personalized treatment for BV. Right now, it's often a trial and error approach, where doctors prescribe metronidazole, and if it doesn't work, they try something else.
Which can be so frustrating and time-consuming.
Absolutely. This test could allow doctors to say, okay, your BV is caused by this specific type of bacteria, so this is the antibiotic that's most likely to work for you.
So no more wasting time with antibiotics that won't do anything.
Exactly. It could also mean less time dealing with symptoms, fewer side effects from unnecessary medication, and less money spent on multiple treatments.
That's amazing. And what about the bigger picture? Does this have implications beyond just individual patients?
Definitely. This kind of targeted antibiotic use is a big win for antibiotic stewardship.
Antibiotic stewardship, that's like using antibiotics responsibly so we don't create superbugs, right?
Exactly. By using antibiotics only when they're likely to be effective, we can help slow down the development of antibiotic resistance, which is a growing problem worldwide.
So this research isn't just about treating BV, it's about protecting the effectiveness of antibiotics in general.
Absolutely. It's a win-win for everyone.
That's fantastic. Now, the research also touched on the idea of using multiple treatments in some cases. What did they say about that?
Well, they suggested that combining metronidazole and clindamycin might be beneficial for some people, especially those with a mix of both resistant and susceptible bacteria.
So like a double whammy to make sure everything gets knocked out.
Kinda. But they emphasized that more research is needed to figure out when and how these combination therapies would be most effective.
Okay, so there's still more to learn. But overall, this new test and the research findings seem super promising. But like any research, it's important to be aware of the limitations. What are some of those?
Good point. One limitation is that the test is currently designed to identify strains with high resistance to metronidazole. It's not as good at picking up on strains with low resistance.
So it might miss some cases where metronidazole might work, but maybe not as effectively as it could.
Right. Also, while they found genetic links to resistance, the exact mechanisms of how those genes actually cause resistance are still being investigated. There might be multiple ways that bacteria can become resistant, which makes it more complex.
So there's still a lot we don't know about how the resistance works at the cellular level.
Yeah, but that's part of the excitement of science. Always more to discover.
For sure. Were there any limitations related to the specific bacteria they studied?
Actually, yeah. They didn't have any strains available to test antibiotic susceptibility for one of the 11 identified genospecies. And one of the highly resistant genospecies was only represented by a single strain in their testing.
So there are some gaps in the data that need to be filled in.
Exactly. Having more data for those specific cases would make the findings even stronger.
Okay, so what about the practicalities of getting this test out into the world? How does it go from a research lab to a doctor's office?
Well, that's another important consideration. Right now, it's still in the research and development phase. For it to become widely available, a commercial lab would need to adopt it and make it part of their testing menu.
So it's not something you can just ask your doctor for right now.
Not yet, but the researchers have filed for a patent, which is a step in the right direction. And interestingly, the health news article we looked at suggests that demand from patients and advocacy groups could actually speed up the process.
So if people make it clear that they want this test, it could become available sooner.
Exactly. It's a good reminder that patient voices matter.
For sure. Now, were there any other limitations with the test itself that the researchers mentioned?
Yes, they noted that the current version of the PCR test isn't great at identifying strains with low resistance to metronidazole within some genospecies. It also doesn't always work reliably to amplify the single strain they had from genospecies number 9.
So there are still some kinks to work out in terms of the test's accuracy and reliability.
Right, but they're actively working on improving it.
Awesome. Now, what did the experts who worked on this research have to say about these findings and what they could mean for the future?
Well, Dr. Catherine Emirati, the lead author, emphasized the potential benefits for patients. She pointed out that this test could help people avoid unnecessary antibiotics, reduce side effects, and lower healthcare costs. She was also really enthusiastic about the potential for faster, more effective treatment and better antibiotic stewardship, especially for people with recurrent BV.
It's great to hear that optimism from the researchers.
Definitely. And Dr. Garth Ehrlich, another researcher involved, provided a really interesting perspective. He emphasized that BV isn't just about one bad bacteria, it's about an imbalance in the entire vaginal microbiome.
The microbiome, that's like the whole community of microorganisms living in a particular environment, right?
Exactly. And he suggested that in some cases, a single antibiotic might not be enough to restore balance, and that combination therapies might be needed. He also pointed out that there seem to be multiple ways that bacteria can become resistant to metronidazole, making it even more important to understand those mechanisms.
So it's a complex ecosystem, and we need to approach treatment with that complexity in mind.
Exactly.
The news article also mentioned something about treating male partners in certain situations. What was that about?
Oh yeah, that was a finding from a different study, but it's definitely relevant.
Yeah.
That study found that for women in monogamous relationships who experience recurrent BV, treating their male partners with antibiotics and a topical antimicrobial cream, in addition to the woman's usual treatment, might help reduce the likelihood of the BV returning.
So in some cases, the male partner might be playing a role in the recurrence of BV.
It seems that way, but more research is needed to confirm that.
Wow, that's a fascinating piece of the puzzle. So we've covered a lot of ground today: the study itself, its potential impact, limitations, and expert opinions. As we wrap up this deep dive, what's the main takeaway for our listeners?
I think the biggest takeaway is that BV, which we often think of as a simple infection, is actually much more complex than we realized. It involves a diverse community of bacteria that can respond differently to treatment, highlighting the need for a more personalized approach.
It really makes you wonder how many other conditions we treat with a one-size-fits-all approach that could benefit from this kind of personalized understanding.
Absolutely. It's a really exciting time for medical research, and this study is a great example of how understanding the intricacies of our bodies can lead to better, more targeted treatments.
So for those of you who struggle with recurrent BV, there's hope on the horizon. This research suggests that in the future, we'll be able to identify the specific bacteria causing your BV and treat it accordingly.
Exactly. We encourage you to stay informed about new developments in diagnostic testing and talk to your healthcare provider about any new options that might be relevant for you.
And if you're interested in learning more about the fascinating world of bacteria and how they impact our health, definitely check out the field of microbiome research. It's a rapidly evolving area of science that's leading to all sorts of breakthroughs in how we diagnose and treat various conditions.
It's a really exciting field.
Absolutely. So that's it for today's deep dive. Thanks for joining us.
Thanks for having me.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.